Okay, so in section 2 of chapter 17, we're really just gonna zoom in on blood and talk about its composition. So blood is a type of connective tissue. If you remember from your tissue chapter in A&P 1, it's one of the 4 main classes of connective tissue that we have. So we had blood, we had bone or osseous tissue as the second main class, a third one was cartilage, and a fourth one was connective tissue proper. But blood was a little bit unique in that it was the only fluid tissue that we actually have in our body. But it's still a connective tissue, which means that we still have our matrix and we still have cells and we still have kind of like a fiber that's present in this connective tissue. The matrix is going to be, again, fluid, and that's going to be what we refer to as plasma. And then the cells, we refer collectively to the cells that are present in our blood as formed elements. When we're talking about fibers, there are dissolved fibrous proteins that will eventually go on to help with blood clotting by forming these fibrin strands, but we don't really have the collagen or elastic fibers that we saw of the other connective tissue types back in that chapter. So a lot of times we only want to look at one of these components of blood, either the matrix or the plasma or the cells or the formed elements. So a lot of times we'll take whole blood, which means blood sample that we've directly taken from a patient, and we centrifuge it. By centrifuging it, we're able to separate our blood into 3 different layers. One of the layers is going to contain erythrocytes or red blood cells. That's going to be at the bottom. And then we're going to have a separate layer, which we refer to as the buffy coat. This one's going to lie directly on top of our erythrocytes. It's really, really thin and small, but this is going to contain both our leukocytes as well as our platelets. And then the top layer, after we centrifuge our blood, is going to contain the plasma. Which is slightly more than half. So about 55% of our whole blood is actually plasma. Anytime you centrifuge something in order to separate them into distinct layers, how they're layered is based off of their density. So the most dense component is going to be at the bottom, while the least dense component is going to be at the top. So here's just an example. Here's our whole blood over here that we came in and withdrew. And we place it in a centrifuge so we can get separation of the different layers based off of their density. Our most dense component is going to be our red blood cells, our erythrocytes, so those would appear at the bottom. We have a very thin middle layer that we would call the buffy coat. This contains both our leukocytes and our platelets. And then we have our plasma at the top. And why is it at the top? Because it's the least dense component. Okay, so just some physical characteristics of blood. It's pretty much a sticky, opaque fluid with a metallic taste. The color of blood does vary based off of how much oxygen the hemoglobin is bound to. So if we have huge amounts of oxygen and within our red blood cells, it's going to be a lot brighter red. So we refer to it as like a scarlet color. If there are low oxygen levels within our red blood cells, it's going to be much, much darker in color. The typical pH of blood is about 7.35 to 7.45. This is slightly alkaline, meaning it's a little bit basic. A pH of 7 is neutral. If it's above 7, it's basic. If it's below 7, it's acidic. So it's slightly alkaline or slightly basic, and it has to maintain this very narrow pH range between the 7.35 and 7.45. It's crucial for a lot of things to happen, which is why the buffers that we're going to talk about later are so important. If we're talking about our body weight as a whole, our blood volume makes up about 8% of our weight. And it is different based off of your gender. So males typically have a slightly higher blood volume, anywhere from 5 to 6 liters, while a female would have about 4 to 5 liters of blood. So let's talk a little bit more about the plasma or the matrix that makes up this type of connective tissue. The bulk of plasma is actually water. So 90% of plasma is water. We also have some dissolved solutes in there, things like nutrients, gases, hormones, which we'll talk about later, waste products from cellular metabolism, proteins, and some inorganic ions. And inorganic just means that these are ions or molecules that do not contain carbon. But the bulk of the solutes that we have in our plasma are actually plasma proteins. And these are plasma proteins always stay in the blood. They're never in the blood to be transported to other cells. They're there and they're produced mostly by our liver. One of the big plasma proteins, or the major one, is called albumin, and this is going to be 60% of all the plasma proteins that we have circulating in our blood. And pretty much it functions as a carrier of other molecules. It also acts as a blood buffer, and it's going to help contribute to plasma osmotic pressure. We're going to talk about buffering of our blood and osmotic pressure later in this chapter, or in chapter 19. So here's a breakdown table of the composition of blood. I do not expect you to memorize the specific description or importance. I really just want you to understand what we can find in our blood plasma. So for example, water, again, that's the bulk, 90%. There's some solutes. These solutes include electrolytes, so things like sodium and chloride ions. They also contain a lot of plasma proteins, and these remain in the blood and do not get distributed to cells. So some of our plasma proteins are fibrinogen, we have globulins, albumin is our major plasma protein, and again, it's really going to help transport and maintain osmotic pressure, and these are going to be produced by the liver. We also have some non-protein nitrogenous substances in there. Basically, these end up in our plasma because of cellular metabolism. So after our tissue cells undergo metabolism, all the waste products get transported in our blood. Where are they found within our blood? Well, our plasma, until we can get them to an organ in which we want to eliminate them in. There's going to be some nutrients, so things that we absorb from our digestive tract are going to enter into our blood and be transported there as well. And as we would expect, our respiratory gases, so both oxygen that we're picking up from our lungs, as well as carbon dioxide that we're picking up from our tissues that are undergoing metabolism. And then hormones, which we'll talk about later on when we hit the endocrine system. So a little bit more on our formed elements, and again, collectively we call the cells that are in blood formed elements. So we have three major formed elements: our red blood cells, and I should not have put RBCs here because you should be used to using the term erythrocyte. Our white blood cells are leukocytes, and our platelets. When we're talking about the three types of formed elements, we only consider one of these three to be a complete cell, and that's going to be our leukocytes, meaning that they have nuclei and organelles, and they can pretty much function on their own. When we're talking about the other two formed elements, our red blood cells and our platelets, they're missing things that make them a whole or complete cell. So red blood cells, they're not going to have any nuclei or organelles. They did at one point, when they were first generated, they did have a nucleus and they did have an organelle, but they eject them at some point in their lifetime, which we'll talk about later. Platelets, these are not actual cells at all. They never were. They're actually pieces that have been broken off of a much larger cell that we call a megakaryocyte. So really, again, of the three formed elements, our leukocytes are the only ones that have all the organelles and things that they need to be considered a complete or whole cell. All of these formed elements, they don't really have a long survival time, so only a couple days until we need to go in and replace them. And when we're talking about where's the origin of the replacement, it's going to be our bone marrow, which you should have touched on during the skeletal system in A&P1. So here's just a picture of a blood smear. It looks much, much better than any of the slides in the lab. But our red blood cells are here, and we also have leukocytes, which typically stay in purple if you're looking at a slide in lab. And there's a 5 different types of leukocytes. So in general, we call them leukocytes, and then platelets. So typically, if you're looking at something that's larger than a red blood cell, it's going to be a leukocyte. If it's much, much smaller than a red blood cell, it's going to be a platelet. So here's a typical blood smear. And again, all of these little pink circles out in the background, those are going to be our erythrocytes. All the really, really tiny things right here, these are going to be our platelets because they're much, much smaller than an erythrocyte. And then we have these larger cells than an erythrocyte, and all of these are leukocytes. But as you can see, we have different types of leukocytes. They're showing you here a monocyte, a lymphocyte, a neutrophil, and a eosinophil. We also have basophils that aren't shown here.